Welcome back to this next video, and in this video we are going to talk about the mutations. Now, a mutation is an alteration or change of the nucleotide sequence of the genome of an organism. When there are changes or alteration in the genome sequence or the nucleotide sequence of the organism, we usually call that as the mutations. Now, this mutation, it can occur in the somatic cells. And when the mutation that occur in the somatic cells, they are not passed to the next generation, or they are not passed to the offsprings. But if the mutations that occur in the gametes, like in the egg or in the sperm genome, those particular mutations that are going to pass to the offsprings. When you talk about the mutations, these mutation, it can be good for the living organisms. For example, by mutations, the bacteria, they are getting resistant to antibiotics, and they can actually increase their population. So those kind of the mutations, they are good for the bacteria as living organisms. But the mutation that can be bad, for example, if you talk about the human beings, and because of the mutations, if cancer is causing, if there is sickle cell anemia because of the mutations, these kind of the mutations, they are not good for the living organisms. So the mutations can be good or that can be bad depending on the effect that have it on the living organisms. If you talk about the types of the mutation, we broadly classify the mutation into two types. One is called is the gene mutations, and the other one that is called is the chromosomal mutations. So first we are going to talk about the gene mutations, what a gene mutation is. Now, as the name indicates, we are talking about the mutation in a gene. So when there is changes in the nucleotide sequence of a gene, we usually call it as the gene mutations. And if you talk about the types of the gene mutations, there are broadly two types of the gene mutation. One kind of the gene mutation is called is the point mutations, and the other one that is called is the frameshift mutations. This point mutation again have got three types. One is called is the missense mutation, the other one is called is the nonsense mutation, and the third one is called is the silent mutations. If you talk about the frameshift mutation, it have got two types. One is called is the insertion frameshift mutations, and the other one that is called is the deletion frameshift mutations. So the gene mutation broadly is divided into the point mutation and the frameshift mutation. So let us talk about the point mutation first. Now, a point mutation is a genetic mutation where a single nucleotide base is changed. So we are talking about a single nucleotide base that is changing. So we are calling that is the point mutation. And of course, we are talking about the point mutation in the genes, about the single nucleotide base change in the gene. Now, this point mutation, as I've told you, it have got further three subtypes. One subtype is called is the missense mutation. Now, when you talk about the missense mutation, this type of the mutation is a change in one DNA base pair, because we are talking about single nucleotide base. So that result in the substitution of one amino acid for another in the protein encoded by a gene. There is a single nucleotide base change, and that base change is actually going to change the amino acid one for another one. So if you talk about the example of the missense mutation, sickle cell anemia is a very good example of the missense mutation. For example, when you talk about the wild type hemoglobin in the sickle cell anemia, when you talk about the sickle cell anemia, the wild type, the normal hemoglobin, if you talk about its DNA, it have got the sequence of CTT. This CTT in messenger RNA is actually represented by the GAA, and when you talk about the protein, it codes for a glutamate amino acid. When you talk about the mutant hemoglobin, where there is a missense mutation, this CTT, this sequence is actually changed into CAT. If you look at this one, the middle T is changed by the adenine over here. So you are actually talking about a single nucleotide change. So when you replace this T by the A, the messenger RNA is changed from GAA to GUA. and this GUA is a codon for the valine amino acid. So instead of the glutamate amino acid in the mutant hemoglobin, you are getting the valine amino acid. And when you are getting the valine amino acid, you are actually having a sickle cell hemoglobin. And when you have the glutamate, you can actually have the normal hemoglobin. So this single mutation over here, a single nucleotide base change, is actually leading to a very fatal kind of the disease, which is the sickle cell hemoglobin. And in this particular case, of course, the mutation, that is not a good mutation, that is a bad mutation. When you talk about the second type of the point mutation, that is called is the nonsense mutation. Now in nonsense mutation, again we are talking about a change in the one DNA base pair, because we are talking about the point mutations. However, in the nonsense mutation, instead of substituting one amino acid for another, it leads to the appearance of a stop codon, where previously there was a codon specifying an amino acid. For example, if you look at this particular sequence, we have got the sequence, the ATG. So this means we are talking about the DNA. So this is ATG, it is coding for methionine. You have got the GAA, it's code for glutamate. You have got the GCA, it's code for the alanine, and you have got the CGT, which code for the glycine. So in normal condition, you are actually getting a protein, an amino acid sequence like this. But when there is a single point mutation, you have got an ATG again, over here you will be getting a methionine. But instead of this GNA, for example, you have got a mutation at this particular point, the G changes to the T. And when the G changes to the T, that means that instead of the GAA, now you will be getting the TAA. And this TAA is actually coding for a stop codon. So you are actually getting a stop codon over here. That means that all of the amino acid below this point, they are not going to be translated, and you will be getting a short protein, which is usually an abnormal kind of the protein and a non-functional kind of the protein. So here you see that a single point mutation is actually leading to the formation of a stop codon. Initially there was this particular codon which was coding for an amino acid, but a single mutation, this codon is changed into a stop codon, thereby all of the amino acid following this point, they will not be translated, and you will be actually getting a non-functional kind of the protein. And if you talk about the example of the nonsense mutation, thalassemia will be a good example of the nonsense mutation. The third type of the point mutation, that is called is the silent mutations. Now, as the name indicates, these kind of the mutation, they are silent. So they do not have any observable effect on the organism phenotype. For example, you have got this codon, the AAA, and you are actually changing the last A into G. So now you are having, after the mutation, the AAG codon. But this is not a problem, because the AAA, it codes for the lysine amino acid, and the AAG codon, it also codes for the lysine amino acid. So you are getting a mutation over here, a point mutation over here, but as both of them, they are coding for the same amino acid, so there will be no change in the structure of the protein, and these kind of the mutation, they are called as the silent mutations. The second type of the gene mutation, they are called is the frameshift mutations. Now, frameshift mutation, that is a mutation caused by the addition or deletion. You can refer them to the indels. When you talk about the addition or deletion, generally it is called as the indels. When there is addition or deletion of base pair or base pairs in the DNA of a gene, which result in the translation of the genetic code in an abnormal reading frame from the position of the mutation to the end. Now this means that because of the addition or deletion, the reading frame that is changed, and at the position where there is addition or deletion, at that particular point and all of the following point, the amino acid, they are going to change, and that change is actually going to lead to the formation of abnormal kind of the proteins. Now this frameshift mutation, it have got two types. One is called is the insertion mutations. Now, when you talk about the insertion mutation, we are actually talking about the addition of the nucleotides. So when the nucleotides, they are added, they actually change the number of the DNA base pairs. An important point to keep in mind about the frameshift mutation, whether you talk about the insertion or the deletion mutations, is that insertion or deletion, that is not a multiple of 3. A multiple of 3 means that 3 nucleotides, they are not added. So the insertion is not a multiple of 3. Let me give you an example of the insertion mutation, how this is actually affecting the protein. Say for example, you have got this sequence, the CAT, 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 CAT. You have got 7 CATs, for example, and this CAT is actually coding for the histidine amino acid. So this is the original DNA code. Now, when there is insertion of a nucleotide sequence, for example, you are having this CAT over here, histidine, the CAT over here, the histidine, the CAT over here, the histidine. Now you are actually adding, say for example, a single nucleotide over here. And when you insert a single nucleotide over here, that means that instead of the CAT, now you will be having the ACA. So you are actually inserting an A at this particular point after the third CAT. So when you add an A over here, I say for example, when you are adding an A over here, that means that the codon now will be the ACA. And when you are getting the ACA, this ACA is actually coding for the threonine. So another codon you will be having is the TCA over here, and this TCA is coding for the serine. Again, you are having the TCA, you will be having another serine over here, you will be having another TCA over here, so you are getting another serine over here. So as you can see is this example that the insertion of a single nucleotide is changing the amino acid at the point where the insertion is taking place. The insertion is taking place over here, so this histidine is changed into the threonine, and all of the following amino acid, they are changed because the codon sequence or the reading frame, that is changing. So these kind of the mutation, they are called as the insertion mutations. And the incorrect amino acids, as you can see over here, you need 7 histidine amino acid over here to make a functional protein. Now you are having 3 histidine, 1 threonine, 3 serines. That means that the incorrect amino acid may produce a male functioning protein, and that particular protein will not be functioning properly. So this kind of the mutation, they are called as the insertion mutation, which is a type of the frameshift mutation. Another type of the frameshift mutation is called as the deletion mutations. When the nucleotides, they are deleted, in the insertion mutation, we were actually inserting the nucleotide sequences. Now in the deletion mutation, when the nucleotides, they are deleted, they are going to change the number of the DNA base pairs. And just like the insertion mutation, a deletion mutation is also not a multiple of the 3 nucleotides. For example, you have got, again, we will be taking the same example. You are having the original DNA, CAT, 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 you are having 7 histidine over here. Now, say for example, you are actually adding an A at this particular, you are actually deleting an A at this particular position. So you are having the CAT, 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 the first 3, and they are very okay. You are getting the 3 histidines over here. Now at this particular point, you are actually deleting this particular A. For example, this A is deleted. That means that the sequence you will be getting over here will be the C, T and C, because this A is deleted and the codon is made up of 3 nucleotides, so you will be having the C, T and C. This C, T and C over here is coding for the leucine amino acid. So you will be having again this C is this CTC over here. That means that the other nucleotide, the other codon, that will be the ATC, and the ATC is coding for the isoleucine. Again, you will be having the ATC, you will be having the isoleucine. So the deletion of a single nucleotide over here is actually changing the amino acid sequence of the protein, and this incorrect amino acid sequence, it may be producing a male functioning kind of the protein. So these are called as the deletion mutations. The second type of the mutation, we discussed that mutation is of two types. One is called as the gene mutations and the other one is called as the chromosomal mutations. So let us talk about the chromosomal mutation now, what a chromosomal mutation means. When we talk about the chromosomal mutation, it is actually a mutation that changes the structure of an individual chromosome, leading to imbalance involving only part of the chromosomes. Now in the chromosomal mutation, that means you are actually changing the structure of the chromosome. You are not talking about the genes, you are actually talking about the chromosome. You are actually talking about the bigger structures now. If you talk about the chromosomal mutation, and if I give you some of the examples, duplication will be an example of the chromosomal mutation, deletion, the translocation, or the inversion, they are the examples of the chromosomal mutations. I'll give you an example of the chromosomal mutation by discussing the translocation over here. Now what a translocation means, this translocation is an example of the chromosomal mutation, and in genetics a chromosomal translocation is a chromosome abnormality caused by the rearrangement of pairs between non-homologous chromosomes. That means that the non-homologous chromosomes, they are actually rearranging their parts, they are changing different parts. The example of the translocation will be the Philadelphia chromosome. And this Philadelphia chromosome is actually a translocation event between the chromosome number 9 and the chromosome number 22. So we are actually talking about the non-homologous chromosome, because chromosome number 9 and the chromosome number 22, they are non-homologous for each other. Now this Philadelphia chromosome is actually associated with the type of the cancer, which is called as the chronic myelogenous leukemia. So that means that this Philadelphia chromosome is actually a bad kind of the mutations. Now what is happening in the formation of the Philadelphia chromosome is that you have got the chromosome number 9 over here, you have got the chromosome number 2 over here. So this is before the translocation. And when the translocation happens, the chromosome number 9 and the chromosome number 22, they are actually changing segments. If you look at the chromosome number 9, this chromosome number 9 have got a gene for a protein which is known as the ABL gene. The chromosome number 22 have got a gene which is called as the BCR gene. So the ABL gene is on the chromosome number 9, the BCR gene is on the chromosome number 22. When the translocation happens, as you can see over here, that the major part of the chromosome number 22, it is shifted to the chromosome number 9, and you are actually getting a very small chromosomes over here. But in this small chromosome, this ABL gene from the chromosome number 9, that is shifted to this small chromosome, and now the BCR and the ABL gene, they are adjacent to each other on the small chromosome, which we call is the Philadelphia chromosome. I hope this is clear. The ABL gene is on the chromosome number 9, the BCR on the chromosome number 22, everything is normal. But after the translocation, this ABL gene from the chromosome number 9, that is shifted to the chromosome number 22, and now the BCR and the ABL, they are adjacent to each other, and we call this particular chromosome as the Philadelphia chromosome, where the ABL and the BCR, they are adjacent to each other. Now, how this particular translocation is actually leading to cancer then? If you talk about the ABL gene, which is present on the chromosome number 9, it codes for a protein which is known as the tyrosine kinase. And this tyrosine kinase, it is a very important enzyme when you talk about the cell division or the cell adhesion, etc. So this tyrosine kinase in normal condition is an important enzyme in the cell division and in the cell adhesion, etc. Now the function of the BCR gene, that is not yet known. What is the function of the BCR? But when the ABL and the BCR, they combine with each other, like in the Philadelphia chromosome, they are now adjacent to each other, they are under the same promoter now. So the ABL BCR gene, when that is translated into the protein, this is again coding for a tyrosine kinase. The normal ABL is coding for a tyrosine kinase. The ABL BCR gene, this combination is also coding for a tyrosine kinase. But this tyrosine kinase is a mutant tyrosine kinase. What I mean by the mutant tyrosine kinase then? In normal condition, when this particular tyrosine kinase, it has performed its function. For example, if it is involved in the cell division, and when the cell division is over, it has performed its function, this tyrosine kinase will be removed from the body, will be removed from the cell by the ubiquitin proteasome system. So when the tyrosine kinase is removed by the ubiquitin proteasome system, that means it will not be able to perform its function now, because it has already performed its function. But when you talk about this mutant tyrosine kinase, the ubiquitin proteasome system is not able to remove this from the cell. This means that this tyrosine kinase is always on. You cannot turn it off. And when you cannot turn it off, that means that it will be leading for the unregulated cell division. The cell will be getting an abnormal signal for the cell division, and when the cell is getting a wrong signal or an abnormal signal for the cell division, it will go for the unregulated cell division, which is actually a property of the cancer. And this ABL-BCR actually lead for the disease which is called as the chronic myelogenous leukemia.